So, my name is Dr. Martin Ronis. I'm from the Department of Pharmacology at Louisiana State University Health Sciences Center in New Orleans. And my task this morning is to talk to you about the animal studies that have been used to suggest that soy and isoflavones might be classified as endocrine disruptors and their appropriateness. So the Endocrine Society position on endocrine disrupting chemicals is that these are exogenous chemicals or mixtures of chemicals that can interfere with hormone action, and these could be either natural or manufactured. They're suggested to impair fertility, thyroid function, neurodevelopment, and increase risk of a variety of diseases, including obesity, autoimmune disease, diabetes, and hormone-sensitive cancers. So the Endocrine Society recommends that these chemicals be treated as having non-monotonic or nonlinear dose responses. They suggest that regulations should protect the most vulnerable populations, which in the case of soy would be infants and children, since the major exposure to soy in the West is through soy infant formula. EDC regulations are suggested that they should use the most sensitive endpoints. And from the perspective of potential estrogenicity of soy, this would include the mammary gland and prepubertal testis. We'll talk a little bit about that later. They suggest the policy should synthesize basic science in vitro animal studies, clinical studies, and epidemiology. And they suggest a precautionary approach is warranted in the absence of conclusive human data. This may be true for a lot of EDCs, but is certainly not true for soy, as we'll see later on in this symposium. So if we look at the generality of the ways in which you test for endocrine disruption, obviously the first thing you do is do in vitro studies to see whether or not said chemical interacts with a hormone receptor. Most of these are in vitro receptor binding studies done with purified receptors in the test tube or using transfected cells in competition with a radiolabeled. In endocrine responsive organs, we would expect to see changes in the physiology of those organs, the growth of those organs, physiological processes such as puberty, the menstrual cycle, fertility. When it comes to sex steroids, we would expect to see changes in gene expression profiles in end organs. We would expect to see changes in cancer endpoints in hormone responsive tissues, changes in cancer incidence and progression in animal chemical cancer models, such as the DMBA model for breast cancer. In more sophisticated studies, you can either knock out the receptor or you can increase levels in transgenic mice. These kinds of approaches are now being extended to other species using CRISPR. But at the end of the day, the most sophisticated endpoints that are being used now involve either gene arrays or massively parallel sequencing approaches using RNA-seq, methyl-seq, ChIP-seq, looking at expression of microRNAs or long non-coding RNAs, or using effects on chromatin using things like ATAC-seq. In terms of soy and the endocrine disruptor controversy surrounding it and isoflavones, the origin of this goes back to studies from 1965 in Western Australia, where it was observed that sheep eating clover became infertile. And this was because of the presence of a phytoestrogen called coumesterol in clover, which has structural similarities to 17-beta-estradiol, as seen here. This led to looking for phytoestrogens in other plants and foodstuffs, and the identification of isoflavones in soy products, in particular genistein, shown here, which, as you can see, also has structural similarities to estradiol. It should be noted that soy foods are complex mixtures, and that in addition to proteins and peptides, there are over 100 phytochemicals bound to soy protein, in this case soy protein isolate, in addition to the isoflavones. These include phytosterols, lysophospholipids, and saponins, and in fact the isoflavones themselves are found as precursors. They're found as genistin and daidzein, which are acetyl-malonyl conjugates, which have to be broken down by gut bacteria to release the bioactive phytoestrogenic aglycones. Further complication is that daidzein is further metabolized to an additional bioactive isoflavone, equol. In vitro binding studies show that genistein binds more tightly into ER beta than ER alpha, and that equol is more potent than as an ER alpha agonist, and that daidzein has weak binding to either receptor. Unfortunately, there are big complications for interpretation of these in vitro studies, largely because of the presence of multiple estrogen receptors. ER alpha and beta can form homo and heterodimers. There are also truncated membrane-bound receptors, and even a G-coupled receptor, GPR30, that binds estrogens. Also, these in vitro studies often lack context because there is generally in vivo possible competition with endogenous estrogens, which you really don't consider in these in vitro studies. When we start to consider the appropriateness of animal models to look at soy and isoflavone health effects, it's important to ask the following questions. So what exactly are we assessing as a potential endocrine disruptor? Are we looking at soy foods, or are we looking at soy extracts or pure isoflavones, which are often found as dietary supplements? This is important because complex mixtures often do not have the same biological properties as pure compounds. And it's also important in terms of appropriate dosing. And there are also appropriate and inappropriate routes of administration. Remember, these are foodstuffs or dietary supplements, and they're eaten. Therefore, studies in which purified isoflavones are being given IV or subcutaneously, which ignore first-pass metabolism, are essentially irrelevant. It's also important to look at relevant developmental windows. As I say, the most exposure to soy products in the West is through soy infant formula, and therefore the neonatal developmental window is an important one to test. Moreover, it's important to look at physiological versus pharmacological dosing. The whole tenet of toxicology is that the dose makes the poison. And it's important that dosing actually reflects the concentrations of aglycones that are found circulating in people after consumption of soy foods or isoflavones. It's also important to take microbiota and first-pass metabolism into account in these studies. Obviously, the microbiota are responsible for generating the aglycones to begin with. They're also responsible for making equol. And first-pass metabolism is extremely important for these compounds. So they can be conjugated with glucuronide or sulfate as they go across the GI. And in fact, only about 5% of the isoflavones that are present in the diet actually are present as aglycones circulating after that first-pass metabolism and conjugation. It's also important to look at the endocrine and immune status of animals in these studies. It's whether or not they have intact sex organs and whether or not the immune system is compromised, as it is in a lot of cancer studies. And there are big species differences, both in metabolism of isoflavones and in terms of the composition of the microbiome, which are obviously also going to make a difference in terms of pharmacokinetics. If we look at species differences in isoflavone metabolism, this is a study we published in 2005, you can see that rats and monkeys are equol machines. That's essentially the major circulating isoflavone, and remember, it's the most potent ER alpha agonist. In contrast, pigs and humans make little, if any, equol. Only about 20% of all humans make equol at all, and only post-weaning. Infants prior to weaning do not make equol. So let's look at some actual studies in which we've tried to take the context of these animal models into account. The first of these is a study we did back in the early 2000s, and this was a lifetime exposure study. In this case, we took spay dolly rats and we fed casein or soy protein isolate from gestational day 4 until sacrifice. The sex organ weight data was looked at in the males. We looked at secondary sex organs, we looked at testosterone and breeding success, and as you can see, we found absolutely nothing. So there were no differences in the weight of the epididymis or the prostate. Testosterone levels were normal, and fertility was normal. Obviously, there are limitations to this animal model. We're feeding soy protein isolate, but exposure is mainly placental and post-weaning because it doesn't get into breast milk. And remember, there are species differences in isoflavone metabolism. So the predominant isoflavone here is equol. Needless to say, equol being the most potent ER alpha agonist, if we didn't see anything there, are we going to see anything with genistein or daidzein? Obviously, this is probably more of a model for Asian populations because we're looking at lifetime exposure. In more recent studies, where we've looked at genome-wide responses in an estrogen-responsive tissue, in this case, the rat mammary gland, and this is in adult overectomized rats, where we've looked directly at the comparison of soy protein isolate with estradiol treatment to restore physiological levels, what we see is very limited overlap. We see 645 regulated genes by estradiol, whereas only 27 are regulated by soy protein isolate, and the overlap is very, very limited indeed, being restricted to 6 genes. When we look more closely at ER alpha and ER beta dependent targets, we see a robust estrogenic response, as can be seen here with cyclin D1, topoisomerase 2A, and kinesin L7. But we see that soy treatment, soy protein feeding did nothing. Moreover, when we combine estradiol treatment with the soy, if anything, what we see is we see a dampened estrogenic response, suggesting potentially anti-estrogenic. More recently, we've looked at the prepubertal rat testis as perhaps the most sensitive tissue. And indeed, we see a